children. We are going to continue our science lesson. In the previous two classes, we learned about vegetative propagation. So, from the branches and the buds, how we can produce good quality saplings and good quality plants we saw, isn't it? So, which are the different methods we learned there? We learned layering, budding and grafting. Now, are these only the methods through which we can produce good quality plants? In these three methods, we learned how new saplings can be produced. So that is part of vegetative propagation. Can we use any other method to produce better seeds? Better quality seeds, that is sexual reproduction. Can we produce better quality seeds? So that is what we are going to learn today. Now in your textbook on page number 13, you can see a picture and the variety description of two varieties are given. Okay, so here you can see tall variety and dwarf variety. Tall variety we know, it's a tall coconut tree. Both we are talking about coconut trees. And what is a dwarf? A dwarf means we know it is a short person, isn't it? Very short person is called as a dwarf. Same way, dwarf variety means it will be very short coconut trees. So the description, qualities of both are given there. What are the qualities of both? Tall variety, it is given 20 to 30 meters high, yields in 6 to 10 years, lifespan is 80 to 100 years, and green colored coconut. Major varieties of these tall varieties are West Coast tall, East Coast tall, Lakshadweep ordinary or Laksha ordinary, it is called. So these are the different varieties which comes under the tall variety of coconut trees. Now look at the dwarf variety. 8 to 10 centimeters high, yields in 4 to 5 years, lifespan is 30 to 35 years, yellow, orange, brown colored coconuts, and the major varieties are Chavakad orange, Chavakad green, and Gangabodham. So these are the different varieties of two tall and the dwarf varieties. Now, what are the advantages and disadvantages of the tall and dwarf varieties? You have to write them down. So look at the advantages of the tall variety. First write down the advantages and write down the disadvantages. So what is the advantage? Okay, lifespan is 80 to 100 years, isn't it? And the color of the coconut is green. Now what is the advantage of the dwarf variety? Yes, it is very short, 8 to 10 meters high, and it will yield in 4 to 5 years. So these are the advantages of both the varieties. What are the disadvantages that you saw here? The first one grows up to 20 to 30 meters high, isn't it? And the yield, it yields only in 6 to 10 years. It will take maximum of 10 years to yield. Okay, so what is the disadvantages of the dwarf variety? Yes, its lifespan is lesser than that of the tall variety and the color is different, isn't it? Different colors are there. So these are the advantages and disadvantages. Now, what will be the quality of the seed which is produced if you join these two varieties? That is a question given there in the textbook. Can we join these two varieties? And can we produce a seed with both the qualities? For that, what method can be adopted there? Can we do grafting, layering, budding on a coconut tree? We cannot do, isn't it? budding, grafting or layering cannot be done on a coconut tree. So what do we do there? We have to adopt another method to produce good quality seeds which combines both the qualities of these two type of coconut trees. We have to get the advantages of both. This method is called as hybridization. What is it called? It is called as hybridization. Now what is hybridization? You would have heard the name hybrid variety. Okay, when you go to buy the seeds, or if you go for a fair in the flower show or something, you would have heard about this is a hybrid variety. So what is the meaning of hybridization? Hybridization is the method of producing new seeds through artificial pollination between plants of the same species but of different characteristics. So what is hybridization? It is the method of producing seeds through artificial pollination. We learned about artificial pollination last year, isn't it? What is artificial pollination? We take the pollen grains of one flower and we pollinate another flower, isn't it, of the same variety. That is, we take it with our hands or using something and we artificially pollinate. There, who is the agent of pollination? Human beings are the agent of pollination. Okay, so that is called as artificial pollination. So here what we are doing? We are artificially pollinating these two trees and producing a new type of seed, fruit, which will have the qualities of both the varieties. This method is called as hybridization. So the seeds formed through this method will have the advantages and disadvantages, and it will be mixed quality. So what do? They take the best seeds out of it and they produce a plant. This method is called as hybridization.
Today I want to talk to you about hybridization of coconut palms. We have, as you can see, a short palm, the dwarf. When crossed with a tall palm, you get very, very good hybrids.
The father is noted through research done earlier to be a very good parent whose good characteristics are transmitted to the progeny, that means the hybrid, and therefore a special mother having a special symbiosis with the father produces this very special hybrid, which is high yielding, good size of coconut, yielding early, having all the characteristics that make for better commercial value for the farmer.
And that's our Sampurna.
The DJ Sampurna is the highest yielding coconut palm, we believe, in the world.
We'll show this to you in stages. The emasculation stage, where a spathe, which is the flower before it opens, is opened manually. Every male flower is removed. Now this process takes 15 minutes to remove over 10,000 male flowers. We mark on the leaf the date of emasculation, and then the technician, who is expert and trained in this and doing it for a long time, he comes along checking this plant regularly over the next 3 weeks, waiting for these female buttons, we call them, to get ready, to get mature. There's a little drop of nectar on the tip of it, which tells them it's ready for pollination. So we have collected the pollen, processed the pollen from the tall trees, processed that in a lab, stored it, and then given it out in vials so that the technician then take the pollen of the chosen variety of tall palm and pollinate the female buttons on this tree. Pollination is done by a brush The brush tip touches the pollen and puts it onto the tip of the little mature button, and the count is taken. 120 days later, we check how many buttons are left. Around 20% are left on the tree, and out of these, around 90% will grow into mature coconuts, which will be our hybrid seed nut, which then goes into the nursery. The nurseryman is an expert. He just has to shake that coconut to know whether the right amount of water is in the coconut. And at the right stage, he will send it to the nursery beds for planting. A month later, germination starts. You see the little sprouts coming out. Within 6 months, the plants are nice and about 2.5 feet tall when they are sold to customers.
Now look at this table here. It is already given in your textbook. The different hybrid varieties of coconut trees. So let us familiarize with some of the coconut trees, hybrid variety, which is at Chandra, Laksha. So what is Chandra, Laksha? Chandra, Laksha is the combination of Vegetative ordinary and Chavakad orange. So when you looked into the first table there, you saw the different varieties of tall variety and dwarf variety. So this is a combination of the tall variety and dwarf variety. So it is called as T into D. What is T? Tall. D is dwarf. So did you understand when you go to buy, they will say this is T into D. T into D coconut. So now you must understand what is T into D. What is T into D? Tall into dwarf. So combination of both. Next one is Chandra Shankara. What is Chandra Shankara? Chavakad orange and West Coast tall. So that is dwarf into tall. D into T. Next is Laksha Ganga. That is Lakshadweep ordinary into Gangabondam. That is T into D. So these are the different qualities produced through hybridization of tall variety and dwarf variety. Now it is not only in coconut that we can do. We can do these in other vegetables also, where grafting, budding and layering is not possible or it is not very successful. We use this method of making hybrid varieties or hybridization. So, good quality seeds of paddies, vegetables can also be developed in this manner. So, there are some of the vegetables which are given there in that table. Look into that table there. You can get many beautiful names there. So, paddy, which are the hybrid varieties of paddy? Pavitra, Hraswa, Annapurna. In P, it is Lola, Mallika, Bhagyalakshmi, Jodika. Green chili, it is Ujjwala, Jwalamukhi, Anugraha. Ladies finger, it is Kiran, Arka, Anamika, Salkirti. Brinjal, it is Surya, Shweta, Harida and Neelima. Then in tomato, it is Mukhi, Anagha and Mukti, Anaga and Akshaya. So you can see those pictures also given there in the textbooks. So these are some of the superior quality seeds which we can get. Okay. So these are some of the hybrid varieties. You have to learn the names of all these hybrid varieties by heart because this will be asked for the exam. Okay. Now, where do we get such seeds? Who gives us such seeds? We know, isn't it? We get it from the Krishi Bhavan. Now, how does these seeds reach the Krishi Bhavan? Who are the ones who does this type of experiments for us? Yes, we know it is through the Agricultural Department of Kerala. The agricultural research centers are set up in Kerala so that they do experiments in such things and they produce good quality seeds and saplings and it is distributed to the farmers so that we can produce good quality and high yielding vegetables and fruits for us. So these are done through agricultural research centers or research institutes which are already there in Kerala. So what are the things that we expect the research centers to do? What do we expect them to do for us? So what is possible in the agricultural research centers? What do they do there? So they try to produce seeds which have high yield, which quickly yield, then they are resistant to infections, low nurturing cost, and availability of quality seeds and planting materials. So these are the things which are taken care of in the agricultural research centers. So what do they give us? What do they keep in mind while producing new type of seeds? The seeds should be of high yield, then they should quickly yield. Okay, the plant should give high yield. They should yield quickly. Nurturing cost must be less. They must be they must be highly resistant to infectious diseases, and we should be able to get it very easily. So that is what the farmers expect the agricultural institutes to do for them. Now, which are the agricultural institutions which are set up in Kerala who are able to do these things for us? There are certain agricultural institutes, research centers in our state. So there are some of them are these: Kerala Agricultural University, Mannuthi or KAU in Thrissur, then Center Tuber Crops Research Center CTCRI, Srikariyam, Thiruvananthapuram, Indian Institute of Spice Research IISR in Calicut in Kozhikode, Rubber Research Institute of India RRII in Kottayam, Central Plantation Crops Research Institute CPCRI in Kasaragod, Regional Research Centers of, so these are the centers, that is agricultural research centers, and also there are regional research centers of Kerala Agricultural University. So under the Agricultural University also there are regional research centers. So these are the main research centers which are working in Kerala to produce such seeds and planting materials, and they are able to give it to us. Now we learned about hybridization and who helps us in producing these good quality seeds also we saw. There is one more method of producing good quality plantlets. That method is called as tissue culture. You must have heard this word tissue culture. What is tissue culture? Tissue culture is the process of producing a new generation from a cell or a tissue of a plant. We have learnt about cell, isn't it? A single cell is taken or a small plant tissue is taken and a new plant is produced from that tissue. Okay, so what is the advantages of doing that? If it is a good plant, or it is a plant which yields good quality seeds, or if it is of a high quality, then the plantlets produced through tissue culture will have all the qualities of that mother plant. So the plantlets with all parental qualities can be developed through this technology. This technology is effectively practiced in plants like plantain, pepper, cardamom, pineapple, etc. So what is the peculiarity of this technology? Hundreds of plantlets can be produced with the same quality of the parent plant. Okay, so if a portion of a parent plant is taken and a tissue is taken, hundreds of plantlets can be produced from a single plant. So that is the biggest advantage of tissue culture, and that plantlet or the plant which is produced through tissue culture will have all the qualities of that plant. So if it is a high yielding, high resistant to disease plant, means all the more than 100 plantlets which are produced will have the same qualities of that mother plant. So Look at this video and understand what is tissue culture.
Tissue culture. In plant breeding, the technique of tissue culture has emerged as a very efficient tool to propagate better improved crops. Tissue culture is much approachable technique than other traditional plant breeding techniques. By using this method, cells, tissues, etc. are maintained and grown in vitro under aseptic conditions in artificial medium, providing necessary nutrients for growth. In 1839, Schleiden and Schwann proposed that cell is the basic unit of life and is capable of autonomy. Each cell, if given an environment, is capable to regenerate into a whole plant. Based on this theory, a German physiologist, Gottlieb Heberland, in 1902 developed the concept of in vitro cell culture, also called tissue culture. Cells in his experiment increased in size but failed to divide, due to which his predictions failed. However, his hypothesis paved the way for more research. He is thus regarded as the father of tissue culture. Taking cue from his experiments, Hanning in 1907 took nearly mature embryos of crucifers and successfully grew them to maturity. In 1908, Simon regenerated callus, buds, and roots from poplar stem segments and established the basis for callus culture. A breakthrough came from White in 1934, who experimented on tomato roots. He reported not only could culture tomato root tips grow, but they could be repeatedly subcultured to fresh medium of inorganic salts supplemented with yeast extract. During 1950s, scientists found that any part of a plant grown in nutrient media under sterile conditions can give rise to a whole new plant. The plant part used in culture is called as explant. This property of plant cells to give rise to a whole new plant is called totipotency. The term totipotency was coined by Stewart in 1968. Totipotency. It requires a nutrient medium that contains sucrose, inorganic salts, vitamins, proteins, and growth regulators, such as auxins and cytokinins. Technique of tissue culture. The first step of tissue culture is excising or cutting a small part of plant tissue, like the leaf, root, or shoot of a plant. This cut-out part, explant, is then placed in a suitable nutrient medium containing a carbon source such as sucrose, inorganic salts, vitamins, amino acids, and growth regulators like auxins, cytokinins, etc. The explants are allowed to multiply and form a group of tissues called callus. Callus is irregular, unorganized, and undifferentiated mass of actively dividing cells. This callus is then allowed to multiply to form many plantlets, which are then transferred to a different container for rapid multiplication. Finally, the developed plantlets are transplanted to the soil for normal growth. Tissue culture is performed using a small part of a plant to develop a large number of plants in a short duration. It is thus also called as micropropagation. As all these plants are developed from the same part of the parent plant, they are genetically identical and called soma clones. Micropropagation is extensively used in horticulture and floriculture and is employed in the large-scale production of tomato, banana, apple, etc. This technique can also be used to propagate high-yielding disease-resistant varieties in a short span. Even in conditions when a plant is diseased, tissue culture is used for recovering healthy plants from diseased plants by taking the explant of the apical or axillary meristem. This is done because the meristem is always free from pathogens due to lack of vascular supply. The process of using the meristem for propagation is known as meristem culture. Culturing of meristems has been successfully carried out in plants such as banana, sugarcane, and potato. There are many advantages of tissue culture, which are as follows. A large number of plants can be grown in a short period of time. Healthy plants can be grown from diseased plants. Plants without seeds can be multiplied. It is useful where sexual reproduction is not possible.
So today we learned what is hybridization and which are the hybrid varieties of the different plants, that is coconut and the other vegetables. And we also saw the different agricultural universities of Kerala, and we learned what is tissue culture. So go through your textbook and answer all the questions which are given. Now, how do we get all these things? These are all produced in agricultural centers. How do we get these hybrid variety of plants or tissue culture plants for our needs or to the farmers? How is it given? It is given through Krishi Bhavan, isn't it? So it is through the Krishi Bhavan that the seed varieties, planting materials developed by the research institutes are given to the farmers, and we also get it from the Krishi Bhavan, isn't it? Each village has a Krishi Bhavan, and these plantlets are distributed through the Krishi Bhavans to the ordinary farmers and to the ordinary people of Kerala. So with this, today's lesson we come to end. Please write down the answers which are given in the textbook.